Right, so hopefully you've got a good idea of the amino acid structure now and how it forms peptide bonds with other amino acids to form a long chain. The primary structure of proteins, and I want to have a bit more of a look at it now and talk about how these the chemical properties of the amino acids actually interact with each other in a way that impacts on the overall proton shape itself. And I think the best way to start, first look at the R group. I mentioned at the start, the R groups. There's a huge variety amongst them, and they can have a big impact on how how the different amino acids interact with each other. They can vary a lot in size. They can vary in their chemical properties in terms of some have polarity, some don't, and there's a huge number of functional groups on them as well. I've drawn a few. Well, I've written out a few over here for us, just so we can get a bit of an idea of them. The simplest R group is on the amino acid glycine. It's just it's just a hydrogen atom. It's just another hydrogen atom attached on. Not polar. It produces a more hydrophobic molecule, and similar with alanine. That's just a simple a CH group added on, and again more hydrophobic really, because there's no polarity in this functional group here. You compare that with the amino acid serine, and you can see here we've actually got we've got an OH group on here. We've got a bit of potential for hydrogen bonding, from the electronegativity of the oxygen molecule there, and so the R group of a serine is going to have polarity to it. It's going to change the way it interacts with other molecules, with other atoms around it. Just to expand on that even further, to make it clear, I've drawn here the R chain of lysine. You can see here we've got a long carbon chain coming off the backbone we've talked about before, and at the end we've got another amino group here. We've got NH3, and in pH conditions that'll be protonated. That'll have a positive charge to it. So again, we've got a molecule here that they're all very different from each other, and they've got a large number of different chemical properties, and will interact with each other in a different way. So we're starting to get an idea how, yes, there's an overall simple pattern to this amino acid, and the R group's going to have a big variety in how they can how each different amino acid can interact, and it's that variety that, as we'll talk about now, allows different sequences in this amino acid chain to have an impact on what the three-dimensional structure will be like. So let's expand on that three-dimensional structure now. We drew in the last video, we saw how the peptide bonds form between different amino acids, and sort of a carbon backbone here. I've drawn it in orange. Is formed, and it's quite difficult to show in 2 dimensions here. But it's not on a single line. This is all very 3-dimensional. We've got groups. Some of these groups will be coming towards us. Like this R group might be coming towards us. The carbon, the oxygen there might be going away. But what's important is that they have a 3-dimensional shape, and it ultimately forms this sort of shape. You've got R groups going in different directions to each other from different amino acids. An oxygen group is going away from each other, and this is also important because it's not just the R groups that have chemical properties, but this oxygen here and this hydrogen here, they have the potential to form hydrogen bonds with each other. I mentioned that before. You have a good idea of what hydrogen bonds are. Nitrogen here, very electronegative, and it causes a bit of polarity, a bit of a positive charge to the hydrogen atom here, and that allows it to be attracted to another oxygen. There might be an oxygen further down the chain that's going to be attracted to. So sort of to summarise really, this makeup that we've got here, we've got a long chain of carbon and nitrogen atoms that forms a backbone for the amino acids, travelling along here, and we've got a huge variety in the R groups, and we've got chemical interactions from the hydrogen, the potential hydrogen bonds from the hydrogen and oxygen coming off this chain. and these are going to be able to interact with each other to form different types of shape. And that's what I'm going to talk about now. I'm going to talk about, why is this important? Why do we want to know about all this? Because these chemical interactions form a certain number of simplistic three-dimensional shapes, and we're going to talk about a few of them now, because there's a few that are very well characterized, and these are called secondary structures here. So secondary structure, and I know we talked a bit about R groups, but secondary structure ones are just formed from these simple the hydrogen bonds that occur between some of these parts of the carbon backbone of the peptide backbone, and these forces are going to interact. And there's two main ones that we should know about, and I'm going to draw them for you now, and they are, the first one is called an alpha helix. That's an alpha, supposed to be an alpha shape there, an alpha helix, and the second one is called a beta pleated sheet. And these are two sort of more advanced. They're fairly simple structures, but they're more advanced than the simple peptide chain that we've talked about so far, and it's starting to develop a 3D shape from very simple chemical interactions between the different amino acids. We'll start with the alpha helix first. Now, what is an alpha helix? Try to draw it now. Basically, if you picture, you've got this, with the long carbon chain I'm talking about, I'll show you up here. This goes on in, it's a long chain, and you've got side chains coming off it here. But this chain doesn't, in the alpha helix, it doesn't just stay in a long chain. It likes to condense itself, and it forms, as you won't be surprised from the name alpha helix, it forms a helix. So you picture here, it starts to coil up and coils round on itself, and it forms this recurring coil. If we picture this line that I'm drawing here is that backbone, that peptide background we talk about. We'll have a carbon, a nitrogen, a carbon, and going round in each other, and we're going to have side chains coming off this. So it might have a side chain over here. There's an R group. We'll have another R group over here maybe, and it forms this shape, which is tightly compacted. And I mentioned the importance of these side hydrogens and oxygens, because they play a role in improving, in stabilizing this structure. They interact with the hydrogen interacts with an oxygen molecule about four further down the chain, and it causes some hydrogen bonding between the two. So there's a bit of hydrogen bonding going on here, which is stabilizing this shape here. It's sometimes a bit difficult to see it like that, but what you can picture it like, it's basically a tube. It's a cylindrical shape like that, with the outside we've got a spiral, a helix going round of the peptide chain. It's condensed itself into a helix like that, and at the top here we've got the chain will continue on, and it might be able to form another alpha helix, and that's the same thing. It's just going to be another sort of another cylindrical shape that goes down. You've got the peptide chain coming around here, like that, and it's all very condensed. And these between the chain, it's stabilized with the hydrogen bonds that we were talking about here. So the second structure I'm going to talk about is as mentioned, called the beta pleated sheet, and it's got fairly similar ideas to the alpha helix. If we'll bring in a few copies of these chains, this is the overall structure again. I think it shows it a bit more easy if I do it this way. And what we, if I overlay them like this, we can see that if you run them alongside each other, then you can picture that the chains can line up and the hydrogen bonds can occur across between the chains. It's not quite done here because there'll be another one a bit further down here where there'll be a hydrogen over here and an oxygen somewhere over here from this chain, and hydrogen bonding will occur, and these weak forces of interaction will occur across the chains. And sort of on a bigger picture view, if we take our imagine this is our chain this is our peptide chain here. Traditionally it's drawn as an arrow in the direction of the chain. You can get these chains lining up next to each other, and they're turning at the end, and another stretch of chain here. It's going to line up to the one next to it, another turn at the end. So this is all just one chain, but they're forming this shape with each other, and in between them all we've got those, we've got the hydrogen, we've got the oxygen forming hydrogen bonds between these chains, and that's stabilizing these molecules. And as we can see here, if you're lining them all next to each other, you get a sheet-like effect, and this is the beta pleated sheet, and that's sort of the second basic structural shape that the proteins can start to form. So this is just the very basic first changes that occur from this simple chain that we've got here at the top, the simple chain of amino acids, and they're starting to form more complex shapes. Okay, so we've started to understand how how this chain of amino acids, this polypeptide chain, can start to form three-dimensional structures. The alpha helix, the beta pleated sheet. Why is this important? It becomes more clear how important it is when you understand that certain amino acids can't form these shapes. So valine, for instance, due to the nature of its R group, I've mentioned how important these R groups are. Due to the nature of valine's R group, it can't form alpha helices. It can't form an alpha helix if there's a valine in that chain. And you can picture, therefore, that we're starting to become important what the order of these amino acids is, because if we want an alpha helix shape for whatever, we've got a big 3D protein structure in mind, then we can't have a valine in that chain of amino acids. And as a summary to this video, I think the most important thing to get from it is that we should be able to start to see now how the order of the amino acids in a protein has an effect on the overall 3D structure. We've got just a linear structure of amino acids, but through the nature of their R groups, through other chemical interactions on the backbone, it started to have an impact on the three-dimensional structure of the protein. And that is what we're going to expand in the next video and say, these R groups are really important in determining the structure, the shape of the final protein, and that impacts on their function and what they do.